All right. Hello, everybody. It is 12 o'clock here on the East Coast of the United States, 9 o'clock back at our headquarters in the Pacific Northwest. So that means it's time to get started for today's webinar. Today, we're going to be talking to you about plant spectroscopy, specifically the uses, the applications, the types of devices that you might encounter, and diving in a little bit more specifically about our CI-710s Spectravue Leaf Spectrometer. Before we get started, Susie's already posted this in the chat. Susie is our host for today. She is our distributor manager. She's going to be the one moderating the chat and the Q&A. But please, if you have any technical difficulties, if I drop out, you can't hear me, or if there's something going wrong with the PowerPoint, please let us know in the chat. But for any questions that you have that pertain to the actual content of the webinar, questions about spectroscopy or the Spectravue device, please put those questions in the Q&A function, and that way I will be able to see them at the end of the webinar, and I will be able to answer those questions and address them. For those of you who aren't familiar with who I am, my name is Galen. I am the Director of Applied Science here at CID Bioscience. I've been with the company for 5 years now. My background is in biochemistry and food science. I have a lot of experience in agriculture, doing quality and safety assessment in the agriculture and the food and the cannabis industries. So on today's agenda, we're going to be talking about just generically what leaf spectroscopy is, the technology behind it, and then I'll go into a little bit more of the applications and the technical specifications of our CI-710s Spectravue Leaf Spectrometer. After that, I'll give you 3 examples of real-world use cases of how this technology has been implemented to improve operations and also help with things like disease detection and other things like that, which we'll get into later. And at the very end, I'll answer any questions that you might have about just generically the application of leaf spectroscopy or our device, the 710s Spectravue specifically. So what is leaf spectroscopy? So leaf spectroscopy is the study of the spectral properties of leaves to assess their health, composition, and physiological responses to environmental factors. Now, in this webinar, we're going to be focusing on visible near-infrared spectroscopy. So this is light in the visible and the near-infrared spectrums, typically from 400 to 2500 nanometers. This is a vibrational spectroscopy approach. So what we're doing is we're shining light into leaf tissue, and that light is then causing vibrations in molecular bonds, typically in OH bonds, NH bonds, CH bonds. And those vibrations cause specific spectral signatures to be achieved, whether you're doing reflectance-based spectroscopy, transmittance-based spectroscopy, absorbance, diffuse reflectance, or interactance, as it's sometimes called as well, and fluorescence methods. All those are different types of optical setups that are commonly used in spectroscopy, the most common being reflectance. So reflectance-based spectroscopy is typically seen in things like multispectral cameras that are mounted on UAVs or satellite imagery. And then hyperspectral imaging is another technology that you might come across that is a little bit more advanced, and that's actually giving you a 2D image as well as the full resolution spectra. So what we're going to be focusing on today, like I said, is visible near-infrared, and specifically handheld tools like the CI-710s Spectravue. So this is a full resolution spectrometer that'll give you all the spectral information in the visible and shortwave near-infrared wavelength ranges. And with that data, there's a lot we can do, as you'll see in a little bit. And so this is a point of contact tool. It's a very small portable tool that you will be using out in the field or in a greenhouse to collect data on a smaller scale than you would with a multispectral UAV setup or with satellite imagery. So some typical applications that we see with using a handheld spectrometer like the Spectravue are applications in the realms of phenotyping. So if you're actually looking for tracking any kind of changes in phenological phases, identification of plants, or even things like if you are doing plant breeding and you are looking for specific phenotypic traits that you are desirable, then you want to actually measure those traits specifically and select for those traits. This is a great instrument for performing those kinds of tasks. One of the most common that we see is things like characterizing stress response. So when you're tracking changes in nutrients and pigments that are caused by any kind of abiotic or biotic stressors. So if you are looking at, impacts of climate change or the impacts of certain types of fertilizer or pesticides, or just salty soil conditions or any kind of abiotic or biotic stressor, and you want to know how that's impacting the health of your plants, then this instrument in spectroscopy in general is a great tool for looking at those stress responses. This also kind of works in the opposite direction, where if you're someone who develops fertilizers, for example, and wants to basically demonstrate that this fertilizer is causing increased plant health or robustness, then that is something you can use this instrument for as well. Another application would be early disease detection, and this is kind of a newer one that has been getting a lot of more traction in the research communities recently. And so typically when you're looking at disease, you are only seeing the symptoms appear visually, and by that point it's too late. And so having tools that you can use to actually observe biochemical and physical chemical changes in plant tissues before you see any kind of visual symptoms is a great tool to have in your toolbox in order to identify disease before it gets to the point where it's too late, and you can implement any kind of mitigation strategies. And then another great application for this kind of tool, for this handheld spectroscopy, is ground truthing. So you can actually use this in tandem with UAV or drone imaging, multispectral imaging, or satellite images to go out and ground truth. So you go out with a higher resolution spectrometer like the CI-710s, and you take samples throughout your field where you already have taken your drone images or your satellite images for your NDVI or some other vegetation index, and you can actually see and compare and either calibrate your images to those ground truths, or just to validate that your UAV drone work is accurate, that your vegetation indices that you're measuring are giving you correct results. So kind of going off that, some common use cases that you might encounter with using spectroscopy. So plant stress measurements are one of the biggest ones, and I mentioned that earlier in the last slide. But with the CI-710s, what we've done is we have actually built in over 40 different vegetation indices. And so with vegetation indices, what you are typically doing is you're measuring either some kind of ratio of a certain wavelength of light versus another wavelength of light. And typically they are at wavelengths where different compounds will typically show increased responses. So chlorophyll A, chlorophyll B, beta carotene, things like that. So plant stress, what you can do is actually look and see visually with the spectra when your plants are stressed using an already pre-made index like the NDVI, which is a greenness index, the photochemical reflectance index, or the nitrogen balance index, which is an index that kind of gives a more robust idea of what's going on with the nitrogen status of the plant. You can also create your own index that you might have discovered works even better for a specific species of plant that you're working with, in order to monitor that plant's health. And so being able to actually go and see, and it's very easy to see, visually sometimes with senescing leaves, but being able to see it on a plant where you aren't getting that kind of visual representation of what's going on as far as stress is concerned, this is a great tool to see where that stress is happening inside the leaf tissue. As far as phytochemicals are concerned, as I already mentioned, you can easily observe changes in chlorophyll, carotenoids, anthocyanins, and many other phytochemicals using the raw spectra output or the actual indices, as I just mentioned. So there are already indices for carotenoid reflectance index, anthocyanin reflectance index, chlorophyll indices, just general greenness indices. So there's tons of other indices that researchers have come up with to look at the levels of these different specific phytochemicals. And so if you are interested in seeing, the specific response of each of these particular chemicals inside the plant, then you can also do that as well. And then another use case, which is a little bit more recent with the advancements in chemometrics and things like that, are nutrient modeling. So you can actually go and create from the raw spectra and analytical reference values from a laboratory. You can create these quantitative models that will actually be able to directly quantify nutrient content in leaf tissue without having to do any sort of destructive analysis. So it's a great tool to use. It does require a lot of extra work up front to be able to build the model itself. But once you have that, then you have a tool that you can take out into the field, and you can measure your nitrogen, phosphorus, potassium levels, other nutrients, calcium, magnesium. You can measure those in the leaf tissue without having to take them into the lab. And so it's an easy, and then at that point, you can actually monitor your plants over time. So the non-destructive nature of spectroscopy allows for increased sampling. So you can go out and take lots of samples because it's a very rapid method, but you can also observe the same plants over time and see changes in those plants based on different treatments or different environmental conditions. You can observe how the changes in that plant are happening on a chemical level, which is a very awesome and great tool to have in your toolbox. Diving in a little bit in the Spectravue itself, I just wanted to point out some features. I've already kind of mentioned a few of these, but with this device, this handheld device, it is a single instrument that you can use to both capture and analyze your data. So you don't have to actually export the data in order to get your results. You can visualize them all on the screen, both the spectra itself as well as the values for any calculated vegetation indices. It's a very easy and intuitive to use instrument. As I mentioned, we already have more than 40 built-in vegetation indices. Those indices are commonly used across many different industries with a lot of researchers having done previous research for them. With those indices, we also like to include any sort of references that might be relevant as far as where we got the index from, as well as a little extra information about, the normal typical ranges for those indices and things like that. So when you actually go to look at your index list that's in your device, you can pull up all that information there on the device in the field. The device as well, I mentioned that reflectance is the most popular, I guess I would say, or most widely used form of spectroscopy right now in the agriculture sector. But this device is able to do reflectance, absorbance, and transmittance measurements all simultaneously. So basically what is happening is that there's a light source in both the top part of the clamp as well as the bottom part. And so what you can do is you actually can illuminate the leaf from two different sides, and we can detect light that's traveling all the way through the leaf, light that's being directly reflected off the leaf, or light that is being absorbed within the leaf. So that's a great feature to have, because having access to all those different spectra will allow you to then utilize whichever form of spectra is going to be the best for if you're modeling disease or if you're modeling nutrients, things like that. Also, additionally, some vegetation indices do require transmittance as opposed to reflectance. And so you can actually calculate those transmittance-based indices using this device. And if you come up with your own indices that are transmittance-based, then you can absolutely do that. It's very intuitive in the fact that all calculations, all the spectra, everything can be exported via USB or Wi-Fi. So if you have some sort of already in place data structure where you want to transmit all of your data to, where it can then be processed, then you can easily do that via Wi-Fi. But everything's also able to be stored locally on a USB drive directly on the device. And then, as I mentioned, we do have the ability to use all this spectral data to perform any sort of advanced chemometric functionalities like PLS modeling or even like neural network modeling. or building your own custom indices. So it's a really open-ended tool that also still has a lot of built-in features. So it's a ready-to-use out-of-the-box tool if you just want to use it for vegetation indices, but it's also very open-ended for researchers that want to do a little more exploration. As far as specifications are concerned, the device is essentially like a tablet where it has a 7-inch touchscreen display. It can be operated at any conditions out in the field, essentially from -10 to 50 degrees Celsius out in the field when you're operating it, and 0 to 90% non-condensing humidity. There is a minimum leaf size requirement. So we do have a 20 by 20 millimeter window optical window that is where we are measuring all of that light transmittance and reflectance. So that you need to ensure that that window is completely covered by leaf tissue. So if you have smaller leaves like blades of grass, or even like conifer needles that you were hoping to study, then all you would have to do is make sure that you are using multiple of those leaves in order to cover the entirety of that window when you're performing measurements. So you wouldn't be able to do individual needles or individual blades of grass if they are smaller than 20 millimeters in width. I already mentioned that it does all three modes: reflectance, transmittance, and absorbance. The spectral range is from 360 to 1100 nanometers. So we get visible all through the shortwave near infrared. The signal to noise ratio is 330 to 1 at full signal, and the resolution is 2.4 nanometers. And as far as the battery life is concerned, approximately 3 to 4 hours in its current configuration if you're using it heavily. And so if you are, only using it very sparingly, your batteries will last a little bit longer. But this is a estimation with, pretty rigorous use is about 3 to 4 hours. You can also program it to either take measurements on an automatic mode, where if you're observing the same plant and you want to get measurements over a certain time period, say you're doing some kind of light studies or something like that, you can have it basically take measurements over a certain period of time automatically, or in the most normal kind of typical mode, it's just a manual measurement. So you just prompt the instrument to measure every time you want to take a measurement. This is just kind of like a 30,000 foot view of the specifications. So if you want more in-depth specifications, you can visit our product page on our website, and Susie will post that link in the chat for you guys so you can actually click it, since you won't be able to interact with the PowerPoint through Zoom. But if you want to see more of our specifications, I would recommend checking that out. And if you have any questions, you can throw them in the Q&A. So let's just jump right into some real world use cases. So this is where I guess people will probably have the most interest in how this instrument is being used. So the first one is a little bit maybe more unconventional than what most people might think. But in this case, what we did was we actually utilized the device to help model moisture content in baby spinach in order to help predict shelf life. And so we actually went out and we did this experiment in-house. We went out and we bought store-bought packaged organic baby spinach, and we stored it in typical storage conditions in the refrigerator, refrigerated conditions, also in non-refrigerated conditions. And we acquired spectral data. So we used the Spectravue, and actually we used 3 different devices to do this data collection. And we would scan the spinach leaves, and then we would take those same spinach leaves that we scanned, and then we would destructively analyze them for moisture content in the laboratory. So we would put them in a forced air oven, and we would basically gravimetrically calculate how much weight was lost due to moisture loss after they were fully dried. And so we would combine those 2 data sets then, the spectral data and the moisture data from the laboratory. We combine those data sets into one database, and then we build a model that predicts then going forward whether or not every time I take a scan, how much moisture is in those spinach leaves. And so what I was showing you in this graph below is a validation graph. And so we actually took this model, and we then, after we built it, we went and we collected independent data scans from a new set of packaged baby spinach, and we scanned all of that. We got our predictions, which are on the y-axis, And then we went with those same leaves and we once again destructively analyzed them in the laboratory to get our analytical or our reference values. And we compared how our model predicted versus what the analytical or destructive method was giving us. And we got a really good correlation of 0.95. The model predicted very well. Because we are using that near infrared range to model moisture content, near infrared spectroscopy is a great tool for monitoring moisture content in tissues, especially plant tissues. So this model worked out very well. So it could easily be used in a lot of these kinds of applications where, say, it's fresh cut flowers or leafy greens or any of these industries where you want to, improve your shelf life so you can have product on the shelves for longer. This is a great tool that you could use to model those shelf lives and be able to tell you and inform you in a non-destructive way. So you wouldn't actually have to lose product after building the model. Non-destructive way to tell you that this product is going to last, for another couple of weeks, or this product is going to go bad tomorrow. So that's a really interesting use case, I thought, for this device that might be a little bit unconventional when thinking about spectroscopy in the ag sector. A little bit more of a common use case, but still kind of more on the, I guess, cutting edge, I would say, of things, is a study from 2019 in which this group was actually modeling nutrients in eucalyptus leaves. And so they were able to use this spectrometer specifically to create these indices that could directly quantify vital plant nutrients. So with nitrogen, potassium, and phosphorus, they had pretty good prediction abilities. But then they also found that in some cases, for some nutrients, this wasn't able to predict nearly as well. So things like the magnesium, that model that the index that they created wasn't nearly predicting as well as they wanted to. The zinc and the iron wasn't predicting as well. So it's really going to be dependent on the type of leaf that you're measuring, the type of tissue, how thick it is, how thin it is. Is it waxy on the outside? Is it not waxy? All those things are going to kind of impact the ability of the models to predict really well for these different analytes. So, but with the nitrogen, phosphorus, and potassium, those are all really important macronutrients that we can now be confident in modeling with the device. So if you wanted to have a device to take out into your cornfield or your soy field or wherever to measure the actual nutrient status of those plants without having to take samples into the lab, if you wanted to get a better representative idea of what's going on in that field, so you could take tons more samples, feel a little bit better about your subsampling plan, then this is the technology that could enable you to do that. So really cool application here back in 2019 from this group. And they have continued off this research for other publications. If you want to check out this publication or others from the author, Susie will post a link to this specific publication, but then you can just look up the authors on ResearchGate or wherever if you wanted to see what else they've been working on. But really cool publication. And then one other big one that I wanted to talk about, this last real world use case here, is modeling disease. And so, as I mentioned earlier, disease is something that we typically only see when it becomes a visual problem, when we can actually see it with our human eyes. But we want to be able to, and we need to be able to identify that disease occurring before it shows any signs of visual symptoms. So what this group actually did is utilize spectral data and machine learning to diagnose diseases in cassava plants. And they were able to get this model to predict disease several weeks prior to the appearance of physical or visual symptoms. And so this has a lot of far-reaching implications for the industry, because especially for plants like citrus, right? Citrus greening disease, being able to identify citrus greening disease way before it's even visually possible to see those symptoms. That's a very important tool to be able to have. And so any kind of other blights or even pests or things like that, being able to see all that before it becomes a actual problem for you and your field and your orchard or whatever, then this is a great tool to be able to use for that. And so combining the power of the spectroscopy with machine learning and AI is going to be a great tool going in the future for doing things like this for disease modeling. And what I wanted to finish and wrap up this presentation on was just some tips and tricks. So anyone that might already own one of these, or if you're interested in buying one, I just have some advice, I guess, for how you actually use this instrument. And so we're going to start off with saying that something that might not be as, I guess, well known. It's in the user manual, but I just want to reiterate this. The instrument needs to be calibrated every time it is power cycled. So if you turn the instrument off and you turn it back on, then before you start taking measurements again, even if you accidentally turned it off in the field, if you turn it back on and you've power cycled it, you need to recalibrate. The calibration process takes legitimately 10 seconds. You just use the dark and light calibration standards that are available in the package that we send you, and you slide those on, and in the menu, the calibration menu, it'll guide you exactly step by step what to do. 10 seconds later, your instrument will be calibrated and ready to go. So please make sure you're doing this. Otherwise, you're going to see drift in your spectra, and that upwards or downwards drift can cause the vegetation indices to predict differently. And then you can't compare side by side anymore, which is the biggest power of this tool, is to be able to compare over time, compare across treatments. So if you don't calibrate, you can't reliably compare those treatments against one another. So please make sure you're always calibrating the instrument. My other set of advice is, this is just might be intuitive to some people, but always carry an extra set of charged batteries with you. The device will come with 2 sets of batteries. Always have that second set charged and on you, just in case the instrument gets low battery and you need to change out and you're in the middle of your data collection, in the middle of the field. You don't want to have to go back and wait and then carry on your experiment the next day or anything like that. So just always have that extra set of charged batteries with you. The last, or not the last thing, but I would highly recommend is always performing measurements in absorbance mode. This will increase the measurement time per measurement, but very, very minimally. But what this will allow you to do is have all 3 types of spectra for every single measurement you do. And just having that, even though you might not think you're going to use it at this time, having that data in your back pocket is going to be invaluable for any work you might want to do in the future. If you go back to look at that data set and you decide you want to try doing some modeling, then you at least have all those different spectral data sets to choose from, as opposed to leaving it just in reflectance mode for just your vegetation indices or just in transmittance mode. This will allow you to actually have all 3 of those spectral data sets in your back pocket for later use. Even if you don't think you're going to use it right now. So it'll increase scan time, like I said, maybe by 1 or 2 seconds per scan, but it's worth it for the extra time, unless you are in an absolute time crunch and you really need that extra, couple of minutes time saving from your sampling. So I would recommend always leaving the instrument in absorbance mode. Another thing that I think is maybe lost on some people is that this device has a pretty powerful capability of being able to back calculate indices. So say you went out into the field and you took some measurements and you only had the NDVI loaded, but you get back from taking all your measurements out in the field that day, you come back and you realize, oh, I actually wanted to calculate a couple of other indices as well. I'm probably going to have to go do that in Excel now manually or something like that. Well, actually, if all you have to do on the device after you've taken all those measurements is go and load those indices, and it'll back calculate all the measurements that you took. It'll calculate the indices for all those measurements. And then when you export your data, all those measurements for those indices will also be exported. So you don't have to do all that manual work afterwards. If you forget to load some indices, just make sure you load them before you export the data. Which is a really nice feature. So it's, for those of us with forgetful minds, then it can be a really helpful feature for us. The other thing would be to utilize the tag feature. So we have seen this in a lot of other industries and a lot of other I guess, non-ag, non-hardware related tech industries where tags are kind of the thing, right? Using a hashtag and then having some sort of metadata associated with it. But using that tag feature will help you keep that data organized. So when you're out in the field and you want to keep your data organized, I highly encourage you to use that tag feature in order to make sure that when you export that data, you know what everything is. You don't have to have all sorts of extra organizational systems in place or anything like that. If you just use that tag feature, you can even on the device have it prompt you automatically with every measurement to insert a tag. So even if that tag is something as simple as saying like P1 for plot one or something like that, then at least that's some kind of extra organization to go into your measurements. While you will always have the date and time stamp, you'll always have the GPS coordinates with every measurement that you take. You still having that little bit of extra metadata is going to help go a long ways with helping you keep your data organized at the end of the day. So I highly recommend using that. And my last piece of advice is to not perform measurements when the battery is indicating low. You can do other sorts of manipulations on the device, like exporting data, selecting data that you want to export, adding indices if you wanted to. That's all fine. But trying to perform measurements when the battery is indicating low is going to result in some, I guess, for lack of a better term, wonky data, because when the battery is low, it's the lamp itself is not going to get the power supply that it needs in order to perform those measurements. And so the lamp will be fluctuating in power, and so that'll cause the spectra to be a little bit off, and that'll cause the measurements, the vegetation indices, to be off. So if your device prompts you with a low battery warning, then that is your signal to stop using it to collect scans, to replace those batteries, to wrap up whatever you're doing on the device, replace your batteries, and then you can continue to take scans after you've calibrated the device. So just please, make sure that you're not taking measurements when the battery is low, and expecting that it is operating in the same way as if the batteries were fully charged or even halfway charged. So those are all my tips, and just wanted to give you guys this brief run through of the different applications that you can encounter using this kind of device. Spectroscopy is a really powerful tool. It's a very powerful tool, but it does require some understanding of how it works, especially if you're trying to do any sort of modeling. If you do have questions about modeling, or you want some advice on modeling or input, we have created actually an entire webinar series on chemometrics and modeling using spectroscopy that might be useful to you to watch. But we're also always available to answer questions on this webinar itself, but also afterwards. If you want to email us with questions about using spectroscopy to model, then please reach out to us. We're happy to provide any insight or guidance that you could possibly need. So this kind of concludes the me talking exclusively portion of this webinar. And I hope you guys learned a little bit about spectroscopy, about the applications of the CI-710s Spectravue Leaf Spectrometer specifically, and learned a little bit about how to use the device and what it's capable of. If you have interest in getting pricing, Susie will post a link to that in the chat. You can follow that link to find more information, request pricing, and just generally, if you have need for the brochure or other sorts of materials to show any of the other teammates, we can send that to you as well. If you want to stay updated with what we're doing, then please go ahead and follow us on Twitter or any other socials. We're on LinkedIn as well. Our website is always being updated with new newsletters. If you're not on our email list and you'd like to receive our newsletters, we always have great content from our newsletters. Really cool cutting edge research happening in the agriculture and plant research fields. So if you want that, you can sign up for that on our website as well. And if you have other questions, you can always feel free to email us or call us, and we're happy to answer whatever questions you have. So thank you for joining us today, and I will go ahead and open up the Q&A function here, and we will go ahead and start with the very first one on the list. So the first question is, in India, land holdings are small, 1 acre to 5 acre. Remote sensing spectroscopy does not work. What is the affordable solution? So the remote sensing referring to the more like UAV work. So if you have small fields or small land holdings, then a portable solution like the CI-710s is a great solution for you because it is a portable device, relatively cheap compared to the operational costs of doing UAV work. And all the analysis is directly on the device. So when you export the data, you don't have any extra necessarily an analysis to do. All the actual data has already been processed. So if you are interested in using spectroscopy, but feel like you don't have the large enough land to justify using remote sensing, then a portable solution like the CI-710s Spectravue is definitely a great solution for you. The next question is, can it be used under tree canopy and how can one do that? So yeah, so actually, if you look at the images from earlier in the slide, and I can actually go back and maybe on my slides here and maybe show the actual image of the device here. So if you look at the device over here on the left, this actually is a leaf clamp. And so what that is going to do is create a closed environment. So you actually are clamping the leaf in between the actual arm there, that silver arm, and the top of the device itself where the optical window is. And so the lamp that's in that arm and then the detector and lamp that are in the actual bottom of the device there will both be able to penetrate light through there in a completely closed environment. So there's no light going to be seeping in or anything like that. Even with thick leaves, even if you were doing something that with a really thick leaf, you're still going to have that closed environment as long as that leaf is covering that entire optical window. So external lighting conditions will have no influence on the actual measurements themselves. So you can use it under tree canopies in open field conditions, in a greenhouse with artificial lighting, whatever, in complete darkness if you really wanted to. Then all those situations are going to yield the same results as far as getting good, accurate spectra because it's a closed system. The next question is, can this test whether a crop is organic or not? That's a great question. So I have not seen anyone use this in a format where they're actually modeling any sort of like pesticide residuals or anything like that. Certainly, it potentially could be used that way, but I don't know of anyone that has used it that way. So I can't say whether or not 100% it would work. Theoretically, it could possibly work, but practically, we just don't know. I don't know of anyone that's used it for that application specifically. Does the calibration drastically impact the data, or will it just be better to do that? So the calibration does drastically is a relative term, but it does significantly impact the data. If you do not calibrate in between and your spectral baseline starts to shift, and over time it'll just continue and continue and continue to shift. If you're if you're doing comparison study, let's say, and you're over the course of 2 months, and you only calibrate the first time you use it, and then you don't calibrate anytime after that. If you actually looked at your spectra on the last day versus the first day from your control plants, then you will see significant differences in where they are at in the height, basically height-wise or bias-wise in that spectra graph. And so that alone will cause artificial inflation or under prediction of those vegetation indices. So yes, making sure that you're calibrating is definitely of the utmost importance. If you, for instance, I guess, have this device already and you have an instance where you forgot to calibrate or you didn't calibrate, and you need to be able to recover that data, there are potentially ways for us to recover that data. And so if that is the case, you can always reach out to us and see if that is something that we can help you with. But it's just best to remember to calibrate every time that instrument's power cycled. The next question is, has the Spectravue been used in viticulture? Are there interesting applications for it in this field? Thank you. So that Spectravue device itself, I don't know of anyone that's using it yet in viticulture, but spectroscopy in general is being used extensively in viticulture, more specifically on the grape side of things. But if you are actually looking to test the health of the plants themselves, then this is going to be a great tool for use in that. I know remote sensing and all those techniques are, being implemented in some areas, but would be a great tool for that. And if you are interested in trying it out, I would encourage you to reach out to us, and we'd be happy to help, run pilot studies or whatever it is that you want to try with the device with viticulture. The next question is, are extra batteries provided with it? And yes, so we do provide 2 full sets of batteries. So you'll be able to have one in the device and one separate. The batteries are also pretty commonly available online. So if you wanted to buy additional batteries, if you just search the same model that we send you, just go to Google and search that same exact model, you could probably find pretty easily some batteries that are located near you that you could get shipped to you. That would work in just the same way. We also provide the battery charger with those extra batteries, so you can always charge your batteries. So the next question is, has anyone ever used the equipment with small leaves that do not cover the whole aperture, for example, conifer leaves? Yeah, so as I mentioned earlier, the thing with the conifers and I guess if you're doing grasses or any kind of other really thin width leaves, is that you have to use multiple. You have to cover the entire aperture with multiple leaves. So that takes away the individual leaf aspect of it. But that is unfortunately the reality with this device, is that you need to be able to cover that entire aperture. So when you try to take measurements and you only have one portion of that optical window covered, then you're going to get some weird spectra, because you're going to have a much different, I guess, or artificially inflated spectrum, because light is just 100% transmitting through on both sides of that conifer needle or that blade of grass. And so it's going to cause some issues with the detector. So when you're using it with the small leaves, you are just going to have to take the approach of it not being an individual leaf sensor at that point, unfortunately. Um, the next question is, hello, is it possible to map a specific tree species in a large area using its spectral signature? If so, how would you go about it? So if I'm understanding correctly, you're trying to map out the entire spectral signature of an entire tree. If that's correct, then in that case, what you would need to use is something like hyperspectral imaging. So even multispectral imaging probably wouldn't cut it in that scenario, because you're only getting a few different specific wavelengths of light. But with hyperspectral imaging, you're getting both that full resolution spectral signature all the way from visible to near infrared, as well as the image, the actual 2D image of that tree. So if you wanted to do that in a 3D way, that's when you're going to the most advanced kind of form right now. So right now, any kind of 3D imaging stuff is usually happening in closed, really expensive, containers that have, very high-tech cameras in them that are giving you a 3D mapped image of that plant. And they can do multispectral imaging as well. But getting hyperspectral and 3D imaging, that's really, really on the cutting edge. And I don't know of anyone that's done anything like that yet. So, I would have to look at the technological universities that are doing the absolute, most cutting edge research and see whether or not any of them have done it. But otherwise, on a 2D basis, you could get multiple 2D images of hyperspectral images, but getting a full 3D rendering and spectral signatures is something that's still kind of not very common, if even possible, right now. The next question is, can we get the recording for the webinar? And of course, this webinar is being recorded, so it'll be sent via email to anyone that registered for the webinar. So yes, this recording will be available once we process it later today or tomorrow. The next question is there an influence of the environment when taking measurements? So for example, sunny versus cloudy days, or even how you're holding the device. So the environment has no impact on the actual measurement itself. Now, so sunny day, cloudy day, the things that could impact it is a wet leaf. So if it rained and you didn't dry the leaf off before you measured it, that external moisture is going to impact the measurement. If as far as holding the device, Yeah, I guess the orientation in which you're holding it will impact the device, because whether or not that optical window is on the bottom of the leaf or on the top of the leaf will alter how that reflectance spectra is being measured. So you can measure the bottom of the leaf or the top of the leaf depending on, as far as reflectance spectra is concerned. Transmittance, it doesn't matter as much. Absorbance will also likely not be very much impacted, but the reflectance will change whether or not you're measuring the top or the bottom of the leaf. And so in that way, the way you're holding the device can impact that reflectance spectra, actually. So great question. Thank you for bringing that up. Is this device pre-calibrated to measure leaf nutrient content? If yes, what crops are calibrated? So it's a great question. We don't have any calibrations on the device for any crops right now. What we have are nutrient indices that aren't quantitative, but they are qualitative. So we have indices on there for nitrogen, phosphorus, and potassium that will give you a unitless measure that you can use for comparison against other crops or other plants that are in different treatments or in different conditions, that will give you an idea of what those nutrient levels are like in comparison to one another, but they are not direct quantitation indices. And so right now, there are no crops that you can use this device for to get direct nutrient content quantitations out of the box. That would require on your end to create the calibration first and then go out and use the device in that way. So the next question is, may I know what is the preferred condition or timing to use the Spectravue? So concerns of abiotic factors that affect the readings at different times. So really, like I mentioned, it is a closed environment. So you're not going to have that much impact from environmental conditions. The biggest things are rain, so wetness on the leaves. You need to make sure that you at least pat those leaves dry before you measure them. Otherwise, that external moisture is going to cause issues. So condensing humidity, right? So we said 0 to 90% non-condensing humidity is our operating conditions. If you have condensing humidity and that optical window keeps getting moisture on it and you take measurements, that's going to impact your measurements. So typically water is the biggest thing that is going to interfere with these measurements. Lighting conditions aren't going to impact it because this is a closed lighting environment. And then as far as other operating conditions, you can pretty much use it in any weather conditions you're going to encounter heat wise. Conditions of light are not going to impact it. So those are the biggest one is always going to be moisture. Time of day, all of those other things are something that you as the researcher are going to have to determine when you want to collect those measurements, because you need to think about, if you're using this as a comparison device across different treatments or something like that, you want to make sure that you're collecting data when everything's, under the same kind of conditions. So when you're doing your data collection, that's when you need to think about, it's not so much how it affects the instrument, it's just collecting data that can actually be used in a comparative way. So don't collect data from, your control plants at night and then the rest of your experimental groups during the day, that kind of thing. So that's where you need to consider those the most. The next question is, can we its use in disease plants for the effect of pathogen on physiological changes in plants? So I guess I'm kind of confused about what this question is asking. This can be used to model disease, to look at changes in plant health status, which would be, if there is any kind of pathogen infection, you are going to see changes in the biochemical and physical chemical properties, which will impact any sort of the spectral signature. Any of the vegetation indices will change, especially the more health-based ones like the nitrogen or the phosphorus, potassium, NDVI, greenness indicators, anthocyanins for stress. So you can use those to actually see those effects of infection, pathogenic infection, before you see physical, actual visual signs in the plants. So that's how I would use it in that case. The next question is, after how much time of fertilizer application, the data regarding leaf contents or nutrition can be recorded? So this really depends on what type of fertilizer is being applied or how it's being applied. Like I mentioned before, any kind of extra moisture or anything like that on the leaves is going to cause problems. Obviously, if you just applied fertilizer and it was sprayed or something like that, and it's on the leaves in any way, that's going to alter your spectra slightly. So you want to give it appropriate time to not be taking scans that could potentially have direct layers of any kind of fertilizer or anything like that on a leaf. So even if it's, just like a mishap spray, it's just, spray going everywhere or whatever, as if you're taking measurements that where there's still moisture or a bunch of residue on the leaf, then that is going to impact the measurement. So I would at least wait long enough to be certain that there is none of that present, and that you can pat that leaf dry without worrying about, removing something that you don't want to remove from that leaf. So that's would be my recommendation. I don't think there's a set time I could give you off the top of my head, but just making sure you have allowed for that. The next question is, any clue on the best protocol to use in case of collecting ground truth for remote sensing? That's a great question. I would say every scenario is going to be different than every other scenario. There's probably not going to be a single best protocol. The most important thing is going to be your subsampling plan, your sampling plan of how you're sampling in order to make sure that you're getting as far as the ground truth sampling, to make sure you're getting a robust idea of the actual, I guess, ground truth of your specific plots. And so coming up with a really robust sampling plan to go out and make sure you're collecting enough samples from different plants to get that idea is going to be the best. But the one thing you're going to want to take into consideration is, especially for remote sensing applications, when you're only really getting any kind of for the most part, top canopy layer images, then what you're going to want to do is when you're taking your measurements for the ground truthing, you want to be taking from the same leaves as what would be measured at from your, or most observed from your remote sensing application. The next question, what could be your advice in using it for spectral profiling of a specific tree species among many others? So if you're just trying to do spectral profiling, I mean, the biggest thing is going to be, like I just said, subsampling and making sure that you're sampling enough of a diverse enough, I guess, locations on the tree, and getting as much sample as you can to make sure you're getting a good average idea of what's going on throughout the entire tree. And then what you can do is actually with machine learning, a lot of different machine learning algorithms, there's a lot of classification algorithms out there, right? So what I would do is once you have those data sets in there, I would look into different kinds of machine learning or matrix algorithms that allow for classification, and allow you to basically identify, say, this is what the spectral profile looks for this tree versus what it looks like for this tree, and have that machine learning model learn about what those different profiles look like and be able to help you then go out and identify in the future. That's what I would recommend. The next question is, I need more details on the indices calculated and their range. Okay, well, that's a great question. If you go to our website, and Susie posted the link to the 710s specifications, you can also go and download our index list. And so we do have the index list posted and available, and it has our lists of all the indices, what their ranges are, because there's 40 plus of them right now, and I don't know every single one off the top of my head. I couldn't just rattle them all off for you now, but that's where I would direct you to get all that information for what is included already on the device. And like I said, you can always input your own custom index as well. Oh, the next question is the same. Can you provide more information on the preloaded indices? Again, I would recommend going to our website. There's a great question. Can you tell a bit more about the use of this device in the cannabis industry? Has there been research published specific for cannabis? So I think the cannabis industry is still just starting to kind of get into the spectroscopy stuff. With this specific spectrometer, with the wavelength range that we have, I can tell you that depending on what the application is, I guess in my mind, I immediately went to thinking of like cannabinoid profiles. So if you are hoping that this technology will replace sending in your samples for triple quad LC-MS, MS analysis to get your cannabis profiles, then I'm unfortunately going to have to report that that's never going to be the case. If you are simply looking for total cannabinoids or just generically THC or CBD, then this technology might be able to actually model those pretty well. The problem with this technology is that when you get to the super trace levels of stuff, it doesn't do very well. And especially when you're trying to separate out based on different isomers or of a single compound, it's not going to do as well with that. So in that regard, that's why I see the potential in cannabinoid profiles being only really used as a kind of overarching cannabinoid category device. But if you are talking more on just the plant health side of things, and it literally can be used just the same way as it is used in corn or wheat or any other crop, essentially, where you're just using it to investigate how your specific treatments or your new soil types, your new lighting conditions are impacting plant health overall. And so I would use those vegetation indices to help provide that information for you. That was a great question. The next question is, can we have a printout of the results directly by attaching with a printer? That's a great question. I don't know off the top of my head, but it has the Wi-Fi capability. So I'm assuming that we could come up with a solution if that was something you needed. We probably could find a way for that to be a reality, to be able to just directly transfer the data to some sort of API that would run that data through to your printer. If you have a wireless printer or a printer capable of interacting with Wi-Fi. Can the graphical representation of data be transferred? That's a really good question. So unfortunately, we don't actually export the actual physical graph that you can see the spectrograph on the device screen there. We do basically what we export is the specific values, transmittance, reflectance, absorbance values for each of those samples that were taken at each wavelength range. And so what you would have to do is when that's exported, it's exported as a CSV. And so in Excel or some other spreadsheet software, you would then just have to graph the spectra yourself, just highlighting those rows and graphing them individually yourself. But great question. The next question is, can I measure thick leaves like cactus? That is a really good question. Depends how thick the leaf is, because light can only, NIR light specifically, can only penetrate so far. So if your leaf is thicker than, I would say, 1 centimeter thick, then I would not recommend using this device because as far as transmittance spectroscopy is concerned, if you are only interested in reflectance, then you can use it on any leaf or really any plant tissue you want, because you're only measuring the reflecting of light off of that surface. If you are interested in absorbance or transmittance, then the thicker the leaf, the more difficult it is for the light to get through, because NIR light is low energy. So the penetration, even though shortwave near infrared is the most penetrative of all the NIR wavelengths, it is still limited in how far it can penetrate. So depends, I guess, on exactly what you're trying to measure. But for reflectance, I would say sure. Anything waxy is gonna likely present a little bit of some interference, but other than that, you should be fine to use it. Thank you for that question, Kendra. Is there a problem if small leaves overlap when trying to cover the area with several of them? So yes, overlapping that's a great, I'm sorry I did not, I guess, qualify my statement earlier with that, but overlapping is going to cause problems if you're doing any sort of transmittance or absorbance-based readings, because you're increasing the thickness of that sample. And so you definitely want to try your best to get a single layer of the leaves on that window. And so to the best of your ability, you don't want to overlap. So thank you for bringing that up. That was a great question. Temperature, humidity, I think I've already covered this, but operating temperatures, the specifications are on the website or earlier in this slideshow. And humidity, as long as it's non-condensing, should not pose an issue. If you have condensing humidity, and it's causing moisture to build up on that optical window, then you're gonna have problems and you're not gonna be able to use the device. The next question is, how often should the calibration be done? As I mentioned earlier in the presentation, you want to calibrate every single time you power cycle the device. So every time it's off and you turn it back on, you calibrate before you do anything. The next question I just answered regarding leaves overlapping. The next is, how complex is it to model the spectrum of a metabolite to determine its contents in a leaf using Spectravue? Any suggested procedure? So yeah, model building, I would say it isn't the least complex process in the world, but it is also one that's very easy to understand once you know what goes into building a model. And I would really recommend that you check out our YouTube and look at our chemometrics series and how to build models. Because there's a lot that goes into planning, especially on the planning side. Before you even start collecting data, there's a lot that goes into it. But what you're going to end up wanting to use is something like you're going to want to have someone that has some kind of data science or programming experience in R or Python, so that you can utilize different features like neural networks or machine learning like TensorFlow, things like that. And so you'll need someone on your team that is capable of handling programming in that way. Other than that, it's really just comes down to planning. But for more information on that process, I would highly recommend checking out that series and then reaching out to us if you have more questions. The next question is, can we update the CI-710s software? Yes, we do provide software updates for free. And in fact, we do have a new software update for current users of the device. So if you currently own a device and you want to update your software, then please reach out to us and we will make sure we get that updated.
I put the link in the chat for the software update.
Awesome. Perfect. Well, then that's where I would go to check it out. And if you own a device already, please update your software. This is a great update that fixes some existing bugs, but also addresses some enhancements to the actual software itself. So would highly recommend. And the last question is, thanks for a great presentation. Well, thank you all for attending. I appreciate you all being here and learning more about this wonderful technology and the Spectravue Leaf Spectrometer. And I hope that a lot of you are thinking about utilizing this in your future operations. And if you are, please feel free to reach out to us anytime so we can talk about your applications and how this technology can assist you in your organizations. So thank you all so much, and I hope you have a great day.